project that Dr. Gray has been developing where he makes this model for in vivo bone formation, and they use fibrin gels. So this experiment has been done with human fibrin and with bovine fibrin, and they have obtained different results according to the fibrin type they use. So this summer they assigned me to do a comparison in the gelation kinetics, the protein permeability, and the cell migration with using these fibrin gels to see if there's any significant difference that would indicate why there were differences in using these fibrin gels in their models. So fibrin gels are formed when thrombin cleaves to fibrinogen. This is a natural response in your body to injury, so it's like the clots that form in your body. Fibrin is very porous and very fibrous, which makes it ideal for cell development and proliferation. So this is why it is commonly used as a scaffold material. So why bovine versus human? So ultimately, in this case, it came down to price. Fundamentally, fibrin coming from bovine and from human should be essentially the same. They should have very similar properties, but based on their studies outcome, we can clearly see that it might not work the same. So utilizing human fibrin can cost 2 to 4 times more than utilizing bovine, and when you're doing research, this price difference can be in between you being able to complete your project or not. So if we look here are some popular scientific suppliers, and we can see that for the same amount of human or bovine materials to make the fibrin gel, there is a significant difference in price. So for my methods, I'm going to be observing the gelation kinetics through absorbency tests, protein permeability using fluorescent recovery after photo bleaching. Some SEM images were taken of the gels to look at their structures and their porosity, and then a cell migration study was performed utilizing rat fibroblasts. And the gels were monitored for 6 hours to determine the movement of the cells on top of the gels. So we targeted 4 different concentrations for the gels. These concentrations are very similar to those ones used in the project that Dr. Gray was developing. So we could keep it, we wanted to keep the concentrations as close as possible to the gels that he used, so these results could be applicable to what he found. 5 gels were utilized for each concentration for each experiment. So and of 5, this was with the exception for the cell migration studies and the SEM images. We didn't need all the concentrations for the cell migrations, and we didn't need as many gels for taking SEM images. So for the gelation kinetic studies, the absorbency was taken at 450 nanometers. This was obtained from literature. We did try 400 and 500 nanometers, which was also used in literature, but 450 gave us a better range of values. So we took the absorbency from the point that we made the gels over a period of time with increasing time intervals. And we did a few practice runs to see at around what point the gels would fully reach polymerization to determine a plateau, and based on that, we did the run for the other gels, and this is just showing the time intervals that I used, and that's an example of the well plate with the gels on it. So for the protein permeability, we use fluorescence recovery after photo bleaching, and this is a method where you're gonna take a fluorescently labeled protein and you're gonna put that into either your gel, your cell, or some type of membrane, depending on what you're studying, and you're gonna expose that protein. You're gonna shoot it with a very high intensity laser until you get a bleached spot depleted of fluorescency, and over time you're gonna observe whatever material you're working on, and the fluorescent protein that is surrounding is gonna come back into that bleached spot, and you can obtain a curve such as this one where you can explore the diffusion kinetics of the material that you're studying, and that's what we did. So for my gels, I use FIT-C labeled bovine zero albumin at a 50% concentration. We mix that with the thrombin, because usually to make the gels, you mix thrombin and then fibrinogen. We use two different lasers, one of 405 nanometers and one of 480 nanometers for both the photo bleaching and the recovery period. We recorded the recovery period for 500 seconds, taking image every second, and we used a gain of 600 and a shutter speed of 700. And you can see here, this was actually the microscope, and if you take a look at this little red circle, you can see the laser shooting at the gel. This is just the interface that we use for the FRAP. It's the REU program, comes with the Leica system. So for the SCM imaging, we prepared the gels, freeze-dried them, lyophilized them, and coated them in silver prior to taking these images. And we took two different magnifications, so 150 and 300. And these are actually my gels. These are the bovine fibrin gels, and this is a human fibrin gel, so you can definitely see there's a difference. So for the cell migration studies, we chose two concentrations based on the protein diffusion results, and those were the 25 units of thrombin to 20 of fibrinogen, and 67 units of thrombin to 13, and that also gives us a very wide range from very little thrombin to high concentration of thrombin. We used two gels per concentration, and they were imaged for 6 hours in 1 to 2 hour intervals. And then we use the ImageJ software to stack the pictures and see the cell movement on the surface of the gels. So based on the gelation kinetic studies, on average, human fibrin polymerizes way faster than bovine. And there's also a less significant difference in the time that it takes to polymerize according to concentration. So you can expect that you can use multiple concentrations and still have very similar gelation kinetics with human fibrin. Bovine fiber gels, the gelation half time was way slower, and we can see here this concentration that takes a lot longer than any other concentration for either gel type. So based on this, there is a significant difference in how they polymerize, but once we looked at the protein recovery, we saw that human fibrin gels had a higher diffusion coefficient, which means that they are better at diffusing proteins. So if you're going to do a study with with fibrin gels that you want some protein to diffuse, human would definitely be a better choice. Here you can see this is just the fitted graphs for the recovery curves of all the concentrations, and the diffusion coefficient was determined using this equation right here, which is derived from the Sompassi equations for the FRAP models, and we use the radius of interest, which is the area that we photo bleach, over the half-time to obtain that diffusion coefficient. So here we see the recovery time for protein, and here is the diffusion coefficient. This is basically just a visual comparison, so it's kind of easier to see, and we can definitely see that there's a faster protein recovery time for the human over here, which is the orange, versus the bovine. And again, the diffusion coefficient for the human is way higher, so your protein is going to be diffusing way readily across the gel. This was seen for all concentrations except this specific concentration over here, 33 units of thrombin to 13 of fibrinogen, which resulted in the slowest recovery time and the lowest diffusion coefficient. So that concentration specifically wouldn't be very effective for protein diffusion. So here's just a little video comparing the FRAP for the 67 thrombin concentration for human and bovine. It's kind of hard to see here because we have a lot of light, but when you're in a dark room, It's more easy to see that on this spot. It starts out very dark, which is the area that we photo bleach, and you guys ideally could see better how it regains the fluorescency. So here are just some SEM images that we took. Human fibrin is way more dense and less porous than bovine fibrin, which might be an indicator of why the protein diffuses differently across these different gel types. So for the cell migration study, unfortunately, I cannot present anything today because we had a little, some setbacks with using the microscope, and I was actually doing this last night, so I couldn't include it on my PowerPoint. But this afternoon, I'm gonna complete these studies and process that data, so if you guys have any questions, you can just go to Dr. Brady's lab later and I might have some answers. But this will further tell us the cell behavior on the fibrin gels. So in conclusion, human fibrin gels have a better protein diffusion and faster gelation kinetics. So applied to Dr. Bray's studies, they would be better off using human for this in vivo bone studies because there's a higher chance that there are growth factors and that the cells will actually grow into those gels. So also from this, you can conclude that fibrin gels from both types are very flexible in terms of how you can get it to behave in the body. So adjusting the concentration, you can adjust how the protein is gonna diffuse across the gel, how fast they're gonna polymerize, so that makes them very ideal for scaffold material, which is actually why it's used so widely. And with the cell migrations, we're gonna see how the cells actually respond to this fibrin gels according to the type. So I wanna give some acknowledgements to the brave program coordinators, which gave me the opportunity to be here, and the University of Texas in San Antonio for hosting me here this summer, to Dr. Bray and Christina Jones, which have mentored me through this whole process. And this is just our grant and some references, and that would be all. Thank you. No questions? Okay.
Sure, go ahead.
Just a question about the kinetics for the duration.
Yes.
So the bovine one, you showed that at the lower concentration it was faster, then it got slower and then faster. Do you have any idea why the kinetics are like that with the concentration?
So we actually, based on different studies that I've read, you would expect for gels with higher concentrations of thrombin to have a faster gelation time, which does stand for most of our gels. So in terms of the human one, we do see that it's kept fairly constant, and once you increase the concentration a little bit here, it goes a little bit higher. But I cannot really say why we, with that gel with a very low concentration of thrombin, we got the highest gelation time. I actually repeated that experiment twice to see if I would get anything different, but that's just how the gel behaved.
Actually, that was the question that I was gonna ask. Although, I think there's a slide or two for me here where it's the, one more. Yeah, here. So that recovery half time, right? The Bovine also shows a weird nonlinear relationship.
Right.
And is that one in the middle the same one that you had the outlier with in the previous slide? It is
This
the same
one right here?
in the lower left, that the 25 concentration.
Mm-hmm.
Yeah, that's the same concentration,
Yes.
isn't it?
Yeah.
That's really odd. I mean, you know, the human one shows this nice linear form.
Right. Yeah, that's definitely something that is very interesting, and it's actually part of why, they assigned me with this project, because they did use bovine the last time that they did the chambers for the in vivo bone formation. And I was actually expecting to get, results very similar to what was used in literature, but it turns out it didn't happen that way. It might have been the combination of concentrations that we use. And I mean, there's definitely a lot of difference when you look at the SCM of the gels, like the porosity and the fiber structures are really altered according to the concentration. So it was definitely something very interesting to look at.
Yeah, I mean, that's a really interesting result, and I think it leads to your point that it's much more difficult to work with this bovine stuff, just because the variability is
Right.
moved from concentration to concentration.
Definitely. I mean, the price difference is very significant, but I would say if you're working with something that you really want it to actually work and you have some protein diffusing growth factors, it would be way better to invest in human fibrin gels, because ultimately your money, would be useful and you could actually get way better results. Yesterday when I was looking at the cells, they were attaching really well to the gels, so that's good. I'm actually a little bit sad that I'm running out of time because I got very invested in this, but hopefully this afternoon we'll have a final conclusion, like, yes, using the human one would definitely make a difference in this model versus using this bovine gel.
Cool, thank you.
Just catching up on a question. I got a quick one. So it's really interesting that you had such a big difference between the human and the bovine. You're lucky, because usually that doesn't happen for us in research, because usually the variables are the same. My question was gonna be, why do you think they're so different? But then you showed the SCM showing that it's a lot denser. So that being said, you're gonna do the cell migration stuff, which is gonna be really important to see whether or not it works in vivo too. There's no right answer to this. What do you think is gonna be the outcome of the cell migration?
Well, based on what I've seen so far, I think from what I've done now, the FRAP recovery studies were the most important. I think the cells are gonna react way better to the human fibrin gels versus the bovine ones. Now, this is my theory, but I have to do a little bit more research on it. I was trying to find yesterday the size of the rat fibroblasts that we're using, because you would think that more porous is better, right? Because the cells have more space to attach. But I think with having a denser gels with smaller pores, we have a higher surface area. So there's gonna be more space for the cells to attach, depending on their size and on the pore size of our fibrin gels, which I did not look into the pore size, but if I had more time, I would definitely go into that. So I think having these very loose structures on these bovine fibrin gels is not really good for the cells. They don't have a lot of surface area to attach and to migrate around, so I do expect to get better cell migration with the human fibrin gels. Do you have a question?